Welcome to the live broadcast of the This Week in Science podcast. I know that countdown music. I just kind of start humming it in my head. I'm like, boom, 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 boom. Yeah, ready to go, counting it down, but also becoming very nicely relaxed. And I'm just going to mellow out here right as the show starts. No, let's do this show, everybody. Are you ready?
Yeah.
So ready.
Let's talk about science, everyone. So, I guess counting down here, huh?
We just did. No more countdowns. Start
What
the
do you
show.
mean no more? Oh, this is like the countdown to like really.
That was the pre-countdown countdown. This is the real countdown.
There's more than one countdown now?
So many countdowns. Sorry to let you know, Justin. Starting this show in 3, 2. This is TWIS. This Week in Science, episode number 881, recorded on Wednesday, June 22nd, 2022. What is crawling on your face? Hey everyone, I'm Dr. Kiki, and tonight on the show, we will fill your heads with meat, mites, and pee. But first...
Disclaimer. Once upon a time in science, there was a question. It wasn't the first question, nor would it be the last, but this particular question had an odd sort of ring to it. One that would echo throughout time and change the way things once were into the way things are now, and it continues to change the world, now and on into the future. It had an odd sort of ring to it because, unlike other questions, this one you could ask about anything. Without changing a word, you could ask it about anything, and the answer to the question, if you followed it through, could change dramatically, depending on when, where, and about what you asked. Once upon a time in science, there was a question, first spoken in a language you have never heard, a simple ordering of words or meaningful grunts, a mere emotive gesture maybe. That conveyance of a post-observational inquiry started humanity down a path that no other creature that has lived upon the earth has been so emboldened as to traverse. Once upon a time in science, there was a question that we here on this show, at the cutting edge of scientific discovery, on the tail end of future modernity, echo each week as it is passed along through scientific study. A question that is now so imbued within the genetic memory of our corporeal existence that it permeates the minds of children still too young to speak it aloud. Foundational to the very essence of our curiosity, the catalytic reaction driving every one of humanity's accomplishments. And still, the most important question we ask today. Once upon a time in science, there was a question. Huh? I wonder why that is. And we will follow the answer to that question across an assemblage of scientific subjects here on This Week in Science, coming up next.
I've got the kind of mind I can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening this week in science? What's happening this week in science?
Good science to you, Kiki and Blair.
And a good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. We are back again to talk about politics. No, we're here to talk about science. Of course, we're here to talk about science, and I'm so excited that you're here with us. We have so many great things to talk about today on this World Day of Camels. Yes. It's Camel Day. It's also Rainforest Day. So let's all be.
And yesterday was Giraffe Day.
Yeah, it's yesterday. It doesn't
That's
count.
yesterday's news.
Yeah.
It's getting a little long in the neck. Anyway, I have stories about eating this week, meat and plants, what your breath says about you, and a bit about an itch. What do you have, Justin?
I've got the wrong run sheet in front of me.
Oh, well, that's not going to work
Wait a
very
sec,
well.
I was
Redundancy,
to tell you about
redundancy.
stories I wasn't going to bring. I've got pea farming. What standing on one leg says about you. Ancient English before the Brexit. And cure for cancer that comes from a plant virus.
Ooh, very exciting. Okay, I'm interested in all these stories. Blair, what's in the animal corner?
I'm gonna have to wait and figure out if that was PEA or PEE, but we will find out.
We will learn.
I brought what everyone wanted to hear about this week, face mites. And then, of course, in the animal corner, I have frogs that are bad at jumping and bats that are good at remembering that also eat frogs. So
Bat frog, bat frog, bat frog,
yeah.
frog bat. It's a new superhero. I'm sure of it. Transitions back and forth. As we jump into the show here tonight, I would like to remind everyone out there that if you haven't yet subscribed to TWIS, you can find us places that podcasts are found. Those places are many and various, but just look for This Week in Science, also TWIS, TWIS. We live broadcast, live stream Wednesday nights, 8 p.m. Pacific time on YouTube, Facebook, and Twitch. We are TWIS Science on Twitch, Twitter, and Instagram. And if all of this is just too much to remember, head over to our website, twis.org, where you can find show notes, links to episodes, and links to subscribe on some of those big podcast platforms. But now, it's time for the science. So let's talk about Sharon. It's not
Who?
Karen, it's Sharon. We're going to talk about Sharon and that red cap that she wears. I mean, where did she get it? Where did Sharon get that red cap?
Who's Charon?
Who is
Well,
that?
Sharon, or a moon of Pluto.
Ah.
Yes. Pluto's
spells it
moon,
with
Sharon, has a nice jaunty red cap at one of its poles. And the question is, where did this redness come from? We first learned of its presence way back when in like 2015 when the New Horizons mission passed by Pluto on its way out into our distant solar system. And the New Horizons mission took some really great pictures of Pluto and Charon and all these outer solar system objects, and everyone saw the data and everyone went, "Whoa, there's a red spot on the pole there, and where did that come from?" And so the hypothesis at the time, based on the evidence, was that because of the extreme swings of Pluto through its orbit around the sun, that there's some extreme seasonality, really, to Pluto and also its moons. So there's this idea that there is hydrogen that gets evaporated from Pluto, goes to Charon. Charon also has a little tiny atmosphere, and that this little atmosphere of Charon also gets kind of heated up, and there is a particular kind of radiation, this light scattering that takes place that they were thinking led to the creation of a red tinted methane product. So basically light impacting molecules in this itty bitty atmosphere as the atmosphere heats up and cools down, And so like whatever was in the atmosphere would kind of lighten up and expand and then condense and rain down onto the pole of the moon. And while in part this is correct, new data that they've been analyzing, they've determined, doing a simulation here on Earth of Charon's conditions, that it's not methane, it's ethane, which is interesting. Ethane is also a little bit less volatile. It doesn't really do a lot of stuff. And so nobody really knows where the red color comes from, because it doesn't make sense, because ethane is normally colorless. And it doesn't normally get a red hue. So it's still a big question, but
it's still the mystery.
It's still a mystery. Nice. Where did the red color come from? Now we have more of a molecular chemical pathway as to like what kind of processes are starting to get it to that point? How do you get the material there? How did it, what's going on there in the first place? And so we're getting a little bit better idea from the research in this study. And the researchers, they've published this in Science Advances, the Journal of Science Advances, and they are from the Southwest Research Institute. And they've used data from NASA's New Horizons mission with laboratory experiments doing, I love this word, exospheric modeling. Because it's atmospheres and things elsewhere, modeling other atmospheres on other things. It's fascinating. I mean, a little frozen moon on the last outpost of the solar system almost. It's got a little atmosphere and it has ethane snow.
Yeah.
What?
It's got this red tint to it, and somebody
And the
looked
red
at it and
tint.
said, "Huh, wonder why that is."
So what I'm understanding is they have basically aquariums, like fish aquariums in the lab. I mean, fancier than that. But then they put
fancier fish aquariums.
dry ice in it, and then just see what color, and then they pipe in different gas and see how it looks, right? I mean, that's what they're doing an artificial exosphere. That's basically all they're doing, right? Is they're just testing gas interactions and what they look like.
Yeah, but they're,
It's so
limiting
fun.
all of the, gas interactions, and then they also were using this. I mentioned the light radiation,
Right.
and so the impact of the photons, and so they also simulated this what's called the Lyman alpha glow to see how that would impact the chemical reactions and what would be happening. Yeah, so they have a special fancy frozen fish tank that
Yes.
they shine lights on.
This is, I feel like there will be science fair experiments that are baby versions of this, right? Where they shine the
Possibly.
UV light through the fish tank full of gas. That seems like a very cool way to go. Find a gas that's reactive to different lights, and then
I love it. Who needs a science fair experiment next year?
Yeah.
Blair just came up with a great one.
Yep.
That's fantastic. Yeah. Anyway, Sharon in her red cap. We still don't know enough about it, so we're going to be waiting for all that juicy gossip as soon as the researchers can publish that. So, moving on, Justin.
What do we got
Pea
here?
farming? You wanted to move on to farming, right? And
yeah. So this is actually
snap peas, sugar snap peas, English peas.
So there's a cowpea. Interestingly, is not the pea fertilizer story. It's the cowpea Mosaic is the virus in a later story. This is human pea.
Oh,
this is
urine.
A team of European and African researchers have been testing human pea as a potential fertilizer. So urine contains phosphorus, nitrogen, potassium. These are the main ingredients of fertilizer. We had a story we were talking about a while ago where they were working with training cows to pee over a grate to collect their urine. Same sort of idea. You could use it as fertilizer.
Train humans to pee over a grate.
Yeah, why aren't we already using our pee to grow crops? Is the big question. If we have this fertilizer that we have readily available, every one of us every day. Well, for one reason, there isn't any form of a pea to farm to table infrastructure in place. Also, oh no.
I mean, how many stories have we had about medications and all the stuff that can be get discovered from human urine? So there's limitations on what we can do here.
But to be fair, that is only gross in the sense that in the best sense of I don't know where my food comes from or how it's grown, sort of a way. Because of course, cow poop, pig poop is a major source of fertilizer. It's not like we're just putting it in putting those seedlings down in sanitary earth and just watching them grow by the miracle of sunlight alone. No, they're normally grown in things that are fertilizers. Right? All right, so this is in their paper published in the journal Agronomy and Sustainable Development. The group described an experiment conducted with farmers in the Niger Republic and the use of human urine on crops. The researchers enlisted the assistance of a group of women living in an isolated part of Niger. These farmers have been struggling to find suitable fertilizers for their grain crops. It's high cost in the commercial products there and scarcity of large-scale livestock to produce manure. So first rule of pea farming club apparently is don't talk about as pea farming. Don't call it pea farming.
Don't use those words.
Don't use the word. Well, apparently
Yeah.
in Nigerian, the common word that you would use for urine is a curse word. It's considered a very offensive word. So they called it oga instead. Oga, which it has some other meaning, but they gave it this name. Anyway, it's still pea. Next, they separated the farmers into two groups. One that ran their farms the way they'd been doing things, and the other group that was going to do pea farming, going to be adding pea to the fertilizer. Over 2 growing seasons, crop yields were measured for both groups. The pea farmers, the pea was provided by the farmers themselves. So they know exactly what's going in there because they know maybe if you're on the antibiotics or something, maybe you don't add it to the mix, right? Maybe if you're on a medication or something, maybe you don't. But they were the ones providing the urine for their own crops. They were taught how to pasteurize, store, and dilute the urine for use as fertilizer. They also added small amounts of animal manure, which was less than the other group, but still. So it was augmented between the two. The data collected from the farms showed that those that had been fertilized using pea produced an average 30% more grain than the traditional farms. 2 years after the experiment, researchers found that more than 1,000 women farmers in the region were using their pea to fertilize their crops. They sanitized. They did have a sanitation system. The urine. Human urine is collected in small plastic containers, transferred to bigger containers, where it is stored for sanitation purposes at least 2 to 3 months before field application. And it was stored under a temperature range 20 to 24 degrees Celsius, which is basically the ambient temperature there. Mm-hmm. And that, according to a paper from 2013, leads to sanitation of sorts.
It's below body temperature, so anything that would have been potentially in the urine wouldn't be able to survive because it would supposedly be surviving in body temperature, but not below it.
Somebody says it burns the poor plants. Good question. So they did dilute. There was a dilution step, which I think was 1 to 1. Just add it.
Seems like just
1 cup of water for every 1 cup of pea.
It seems also, though, that we do know that the ground is like a filter, and also the sun kills bacteria and other things that can be dangerous. So, I don't know. It seems like putting the urine in the sun would be a good sanitation step as well. But maybe this is very fascinating to me. I want to know why it works.
And Justin, where was this again?
Nigeria.
So that's another thing I think is interesting is I want to say in a lot of countries besides the United States, people wash their fruits and veggies much more thoroughly than we do. They'll actually dunk them in diluted bleach a lot of times. And so I am curious about how that process worked and if that was part of the success of this process. Also because just the ick factor, right? Like if you know you're washing things that well, you might be less perturbed by it. Let's not take a minute to think about in the United States that we don't wash our stuff super well. They're washed by the farms, but we don't wash them in our kitchen sink to the same extent, and we still have raccoons and feral cats and all sorts of things that are peeing all over our produce. So.
We do. Although
and
not
that's
in
where
your grocery bags, hopefully.
And that's where it's because this is fertilizer, so this is getting applied a couple times during the growing season and not onto the plants. It's not like to give the
au naturel application.
No, but I mean it's being applied basically to the soil.
Yeah.
I need to get it for the roots of the plant before it grows the produce or before the grain, right? So it's not like you're spraying it on the grain. That would be too late for a fertilizer.
Right.
Well,
I'm all for anything that prevents us from putting urine in a bowl full of fresh water because that is a waste of water. So if this can divert that would be great. Whoever came up with that, I'm mad at you. That was a bad idea.
I'm not. I'm not.
Letting it run
Whoever
out to the river and tributary system.
That's the greatest invention.
I feel like there are other things out there that we could be doing to reduce smell that wouldn't involve wasting a bunch of fresh water.
Yeah, but then like wherever you are,
gray water
There
recycling.
is.
Yes,
there is. But here's what you would have to do, Blair. You and probably all of the other residents on your block would have to leave your home each time, walk over to, because you got to have it some distance from the house, to the pit that was dug. It's got the nice little toilet sitting over the top of it.
You're telling me in 2022 there's not some sort of technology that we could come up with that would reduce the smell of urine, that would filter it on its way out, that wouldn't require gallons of water? Because I'm pretty sure there would be. That's just
there is, but again, then you're talking about the slide that has to be south facing, so with the glass over it, so the sun can come and dry it.
I think
you're not getting rid of the smell.
Anything potentially sustainable and putting us in that direction that we can start thinking about putting on our radar for future development.
But gray water flushing would make all the sense in the world, and we don't do that enough. You're right.
Yeah. Oh, after all this water talk, though, I'm thinking about maybe I need a cup of tea.
Yeah. Would you like some arthropods with your tea?
No. What?
Researchers from Trier University and the Max Planck Institute for Evolutionary Biology wanted to look at eDNA in commercially sold tea and dried herbs. eDNA, that's environmental DNA. It's left behind in natural environments by creatures like spiders and other arthropods. And this is usually when you're talking about eDNA, you're not talking about crushed up actual arthropods. You are talking about their saliva, their leavings, speaking of pee, and poo, all this kind of stuff when they crawl on the frass. Exactly. We try to give it a different name so it's less gross, right? So they crawl on these surfaces, they chew on these surfaces, they leave behind eDNA. And just like we talk about eDNA in the oceans from fish, that's also often from peeing. This is a similar measure. And so this was a kind of a proof of concept that they could test tea and herbs, because they know that the two best ways to study eDNA is in some sort of water or in a preserved, super dry location. Those are the two best ways to get eDNA. So they went, what about tea and herbs? And they found eDNA for over 1,200 unique species of arthropods.
Wow.
Yes, this is in 40 tea and herb samples, just 40 samples, 1,200 species. They found evidence of 3,264 individual arthropods spanning very different types: predators, herbivores, detritivores, parasitoids. They found that every sample, if you picked one sample, it contained eDNA from at least 200 animals. And the one that had the most?
That's significant.
What kind of tea do you like to drink, Kiki?
I like black tea.
Okay, Justin?
The same. It's
okay, you're both safe.
I'm an Earl Grey girl.
Yeah, me too.
Green tea had the most. Green tea.
Oh, the bugs like the green tea, huh?
Yes,
You'd
leafy.
think there's a very bitter flavor to most green teas, though. You'd
Mhm.
think that would be pushing the insects away, but
Yeah, I don't know.
interesting.
So this research shows that the range of eDNA in studies can be expanded to pretty much anything that is preserved or dry. And so looking at actually commercially sold items could help them to look at the ecology of specific areas if you know where those things are sourced, all sorts of good stuff like that. And so this is a potential way of monitoring those specific ecosystems. Of course, this could provide useful as global warming continues. Does that eDNA change? Do the arthropods visiting that area change? And it can track other impacts like pollination and all sorts of other things. So eDNA from, in this case, food items can help keep a track on specific ecosystems.
Oh, that's interesting. Yeah, that's a wonderful application.
Yeah. So think about that next time you're having some tea.
Yeah.
I would love to find out what the different insects are using. I mean, this is their ecosystem, right? Even though but most of these teas are, I mean, I don't know. Are the teas in monoculture? How are the
Great question.
insects using them? So if teas are grown in more complex, diverse environments, which I know some growers do that, does that mean that there's a greater diversity of insects? If it's monoculture, is there a lower diversity? And then how many of these insects are biting and chewing on these plants, which have a lot of these antioxidants, which are also, those are compounds that are meant to protect the plants and be, you know, trying to get the insects to stop biting them. And then, and how many of them are just landing there and it's a place to put a cocoon, or it's a place to have, a little evening away from the kids.
And that's sort of how I would picture it, because there's when you're drying all of this, it's not like there's an indoor factory sanitary floor lab where they've got these outside or in barns, and it's just throw all the leaves in there and out there and lay them out and let them just dry in the sun and the air. So the bugs are going to be everywhere.
Yeah, a lot of, I mean, that's the tea leaves, usually they are just dried out places. Sometimes they're, Blair, you've seen the inside workings of the, what is it? The
Celestial Seasonings plant.
The Celestial Seasonings plant. So did you learn what they do with their herbs?
I mean, there were lots of conveyor belts. It was many years ago now, actually.
Yeah, it was a while ago.
I do remember going in the peppermint room because it was
Yes.
very strong. And I also remember being told that herbal tea is not truly tea because it does not include any tea leaves. So
Interesting.
chamomile is not tea. Sorry, everyone.
I'm still gonna call it. It's an infusion.
There's still bug DNA in it, though. Guaranteed.
And so Goldizator over in Twitch is asking about the aging of the tea broke down the DNA, and not any more than just any regular outdoor process would. So there's probably a lot that is not preserved, but not destroyed either.
Yeah, well, it's one food product that you can't wash in hot water before you hand it over to the public.
Right.
True.
It would ruin the tea.
Yeah.
Exactly. It would ruin the whole purpose. Moving on from pea to tea to meat. Let's talk about the meat that we eat and its alternatives. Have you ever wondered whether the protein, the meat that you eat, is whether it's treated the same by your cells as regular meat, like meat versus tofu versus wheat gluten versus, like, have ever wondered whether these different proteins get the same treatment from cells, or whether there's
so I would assume that my veggie burger is going to be as useful as if I'd eaten vegetables and not meat. So
but that's a veggie burger. That's not an Impossible Burger. That's different.
Which is a meat alternative, right? Something that is specifically supposed to be a chicken substitute, for instance.
Because
Right,
there's protein in mushrooms. So that's a question. If you eat a portobello mushroom, is that considered the same by your body as the grams of protein that you would get from a teeny tiny bit of steak? Which my guess would have been that the difference is in the type of other nutrients you absorb at the same time.
Yeah.
Right. And so like you talk about nutrition, you say you need a little fat to help with the processing of protein, and you need omega-3s, and you need all these different you need fiber, you need all these things in concert to get the optimum benefits of these items. But I've never really thought about whether, if you're just looking at protein, if it would matter where it came from.
Well, some chemists decided to check on this, and they are publishing in the American Chemical Society's Journal of Agricultural and Food Chemistry their analysis of how cells basically absorbed and dealt with chicken protein or a meat substitute that they created out of soy and wheat gluten. When they took this mixture, that was not a commercial on the market kind of mixture. This is something that they created. They created a model meat alternative out of these products. And then they used an extrusion process. So it ended up being this kind of long, fibrous, filamentous meat substitute, very similar to chicken, very similar to muscle cells and the way that they are situated within the muscle, long and fibrous. Anyway, they cooked them, and they cooked pieces of chicken. They then ground up the meat substitute, and they ground up the chicken. And then they put that ground up chicken and then the ground up meat substitute in an enzyme that humans use to digest food. So an enzyme that's
no
found
one's
in our...
eating these. Okay.
They weren't eating them.
As,
No.
as you kept
No,
going,
this
I was
is
like,
a-
that sounds less and less yummy.
It wasn't
So
very...
it's a food science lab.
They-
So they've
well,
got
in a lab,
they've
they
got a
created
faux
the thing.
digestive system.
Okay,
Yeah.
all right.
They're
I'm with you. I got it.
that they do
Simulated
chemical analyze
digestion.
after.
Yes. So they simulated digestion in a lab, and then they basically they put these digested meat solutions in with cells in petri dishes, and they determined that the meat substitute proteins were not absorbed as well as the chicken proteins by the human cells. And so the question is, what's going on there? And so, Blair, I'm to your point earlier about how things are put together. All these researchers did is take soy and wheat gluten and create a meat-like product that doesn't go into all the other stuff. What about fats? What about the other nutrients that are in chicken? What other stuff is in a slice of chicken that would make the cells more likely to incorporate them, to endocytose them? What would make those proteins more useful? When you would think a protein is a protein is a protein, right? But they're not. So what is the difference, and how? Obviously there is a difference.
Yeah, my money's on that it's like the stuff in the chicken.
Yeah, so we don't know. So is it stuff in the chicken? Is it some aspect of the initial protein structure, and then how the different proteins cook because of the amount of water and fat that was in the different substances that led to the eventual shape of the proteins that made them less or more likely to be absorbed? But this is a study. I don't know, but it's not exactly the same as having your fake chicken nugget versus a real chicken nugget. I mean, it's not exactly the same as that, but we're not there yet with that understanding. But this at least is an interesting piece of the puzzle.
Yeah.
What makes the fake meat different from the real meat?
What would really make that interesting is if we found out it was like the fat, right? And then one of the things that fake meat is supposed to be promoted for is that it's healthier for you. So if we have to make the fake meat less healthy in order for the protein to be better for your body,
Well, yeah, more useful.
my
Yeah.
God, what are we doing?
What are we doing, people?
I'll tell you one thing, that fake meat,
Mhm.
there's not a whole lot of environmental DNA from insects.
At that point, probably not.
The
Although
chicken, but maybe the soy has it.
Although I bet you it does have environmental DNA from whatever organism was producing it.
Yes, certainly. Has normal DNA.
That too.
I don't know.
That too.
But moving on from DNA, I mean, we want to be healthy, right? We want to live long lives. So, should I just stand on one leg?
Yep.
Is that
Well,
just-
you
that's what
scooped
it
the
is?
whole story.
I'm doing it right now.
You scooped the whole story. That was my
You'd
big reveal at the
never
end. I was
know.
going to do all this lead up, and then it was going to be stand on one leg, and that was the big ending. Well, anyway, this is good news for people who like to stand on one leg, because that ability to stand on one leg for just 10 seconds cuts in half the risk of death from all causes, according to research published
Kiki's
in
showing
the
off.
British journal Sports Medicine, suggesting that a simple, safe balance test could be included in routine health checks. Oh, I hope not. They've got so many ridiculous things. When you got okay, so
the
the
jiggle pose. Sorry.
hey, I
I'm
got a
doing my
question.
yoga now.
Yeah,
How do they? Oh yeah, no, yoga in a chair. You're just gonna
I'm standing.
wrap it up like this, and then with one leg.
All right. Okay. So what's going on with
So-
this story? What does this mean?
They're going to make people stand on one leg now to see if they're healthy, which you don't turn your head and cough to check for hernias anymore, I don't think. But it's a thing guys had to do. They would grab you and they'd say, okay, and they'd turn your head and cough. And the reason you did that is so you didn't cough on the doctor, but they were looking for hernias. Like there's other ways to look for hernias. When they listen to your heart, do you know how many doctors can actually pick out a heart murmur or an arrhythmia just listening to your heart? It's none. They can listen to your lungs just fine, but they're not that good. Your doctor's not trained that well. They do a lot of things. Now they're gonna have people standing on one leg, falling down. Well, at least they're already at the doctors. So
Doctors
unlike-
listening to TWIS, direct your emails to Justin.
Well, actually, no, but I think actually doctors are why we don't do that anymore. Like it was this, we had all these weird practices that doctors would do in a checkup that at some point they're like, I've never found a hernia. I've been practicing medicine for 30 years, like making people turn their head and cough while I grabbed them by their genitalia. Didn't find one. Anyway, no hernias that way.
But
Anyway,
why is this going to be more successful
trying to
than
get
the
to that.
coughing?
I keep trying
This
to
is
get
going
to
to be
that.
more successful. Why?
I don't think it is. Unlike aerobic fitness, muscle strength, flexibility, which wane quickly as we age, balance actually tends to stick around reasonably well until about the 6th decade of life. Then it starts to wane. Oh boy, does it wane rapidly, say the researchers. Balance assessment isn't routinely included in middle age and older folks, so there's no standardized test. So they kind of created a standardized test for this. This was all set up back in 1994 to assess associations between various measures of physical fitness. This is part of the ClinMEX exercise cohort study. The one-legged study included 1,702 of those participants, aged 51 to 75, with an average age of 61. As part of this checkup, participants were asked to stand on one leg for 10 seconds without any additional support. And they had to keep their arms at their sides, look forward, and I think they had to have the free leg behind the calf of their other leg.
Oh.
So they couldn't do bow pose. Like I can go out there and bow pose and hold it probably longer than I can do that weird posture that they're talking about.
I'm trying to put my leg behind my other leg.
Yeah,
I have my arms down.
Yeah,
It's a
at
very-
least let me go flamingo. Come on.
yeah, no, that would be, but they said because they need everybody to be able to do it, they tried to make it as simple of a one-legged test. And in fact, you could
my-
pick a leg and you had 3 attempts on either foot until you could get to 10 seconds, and then it was a pass-fail. If you just couldn't do it in any of those attempts, it was a fail. If you even got 1 out of 3, you passed the thing. All in all, those people who were, let's see, what is it? 51 to 55, were about 5% couldn't do it.
Mhm.
In the next 5 years, it was 8%. Between 61 and 65, 18% suddenly could not accomplish this task. Just under 37% amongst those 66 to 70 years old. And over half, 54% of those aged 71 to 75 were unable to complete the test. When they looked over their data and stuff, what they found was the chances of dying of any cause. You get cancer, people died of heart congestion problems, they got the COVID, hit by a bus, drowned while snorkeling off the coast of Hawaii, however they died. The chances of death, the 10 years after failing that test,
Mhm.
increased by 80-something%. Oh, I lost my number.
Yeah, but
84%.
so it increases massively if you can't stand on one leg for 10 seconds.
Yeah. So when I first read this, I was like, well, of course, the 75-year-olds are the ones doing the worst at this test. So you look at all the
They're
numbers.
going to be living to 85 or,
Right.
yeah.
The chances of making their next 10 years much tougher than the 55-year-old making his next 10 years, right? So that seemed weighted. But according to this, they weighted for all the factors they could, including age, and they still got to 84%.
Mm.
Yeah, so it's probably related to neurological factors.
Mhm.
It's probably related. I mean, there's probably just general health factors involved as well.
Yeah, in general, it says here those
yeah.
who failed the test had poor health, higher proportion
Mhm.
were obese and or had heart disease, high blood pressure, and unhealthy blood fat profiles, as well as diabetes was common.
Mhm.
38% of those who couldn't do it had type 2 diabetes. So they were 3 times more likely, that group. But again, that's something that can also hit you hard or hit you later in years. So.
Right.
But this reminds me, there was another study like this that I'm cognizant of, and every time I'm walking around now, there was one almost exactly like this, and I'm wondering if they used the same data because it was the same like chance to die in the next 10 years of any cause, which was the rate that people walk at, how fast you walk, was related to how much time you had
Mhm.
left. So the slow walkers, they found, died quicker than the fast walkers. Fast walkers, it means you got a lot of life left in you. So if you can walk fast and stand on one leg for 10 seconds, you're fine. No need to go to the doctor.
You're
no matter what
great.
they-
Yeah. Yeah. Well, I mean, it really does reiterate the importance of doing whatever you can do to maintain the best health that is possible for you
Mhm.
and maintaining activity as late in life as is possible, just as much as you can get will help with that general health. And yeah, test yourself every day if you can. Stand on one leg. Make sure you can do
I
it for 10 seconds. Sit
can't
down in a
believe-
chair, stand up, stand on one leg. There you go.
Can't believe old man Jackson passed. You know, he was such a fast walker and he could stand on one leg. Yeah, but he drank heavily and smoked a carton of cigarettes a day.
Right.
Oh, yeah. But did you see him move around? He
But
was
he can stand on one leg better than a flamingo.
Yeah.
Oh my goodness. Well, as we're on the being healthy tip, one thing that
Mhm.
you can do to be healthy for yourself can also help the climate. A new study published in Nature Food estimates the carbon footprint of the global food transport system. And it's fascinating analysis of emissions from the entire food supply chain. So it's got they're looking at emissions from the transport of fertilizers, like the animal fertilizers we were talking earlier, machinery, the feed for animals, like the cattle that become our food, and the moving of food itself. And they found that in 1 year, global food miles were responsible for 3 billion tons of carbon dioxide equivalent emissions. So that includes carbon dioxide and other greenhouse compounds that have effects. So 3 billion tons. And this is larger, significantly larger than previous estimates. Now, there are some people who are still debating, as always happens in science, debating the estimates that have been made in this study because some people contend that they are overestimating certain aspects of the emissions and how much fuel is used, like for train transport of coal, for instance. In the United States, coal is transported on trains very often and not on trucks. So trains are going to be a little bit better on the emissions than trucks on the road are, slightly. Anyway, there are all sorts of details, and it gets very complex when you really dig into the food systems of our planet and the global supply chains and how we use them. One thing that I thought was very interesting was their attempt to look at for instance, the entire supply chain of something like red meat for the country of China. Chemicals for fertilizers for the feed for the cattle are produced in Canada and in North Africa. Those go to support the growth of vegetables and fruit and soybeans in the United States and Brazil. Additionally, those are then transported to China, where the livestock are grown, to be able to feed the livestock there. Coal is produced in Australia and Indonesia that is transported to China for to power the manufacturing process for the sale of the red meat to the Chinese citizen. So it's an incredibly long process, and each aspect of the process has its own inputs and outputs. So the final point to this story, however, is not just, oh, let's all be sad about all the difficulty of the human food system and the web that we have created for ourselves, but rather we can take a picture of how food and energy are emitted and used, or food is used and energy emitted, the carbon dioxide is emitted around the globe. And what it tells us is that plants are more often grown and shipped around the world, and plants can be something that can be grown more locally and have less of an impact on these carbon emissions. And especially richer countries, if we reduce as we've said lots of times, our reliance on meat eating, that would reduce a big aspect of these emissions. But not just eat more plants, but eat more plants grown locally. So eat more seasonal produce. Don't buy things from Brazil, or even though this is a global economy and we have the money and we can buy whatever we want, whenever we want. If you start shopping from a more local seasonal mindset, it has a huge benefit, or it will have, if at the population level, people make choices like this to be able to shop locally.
The more you buy local, also the longer the food lasts, which
Yes.
reduces contributions to food waste, which also reduces the need to buy more food, which also feeds this whole system. So that's, yeah. Is that the chime of being correct? I just got that.
Da-da-da-da, you are correct.
Winner. Yeah, so
You
there's
have
that.
one.
There's also, it's if you're shipping food, there's a better chance it's being preserved in some ways, which refrigeration I think is the number one cause of carbon emissions according to Project Drawdown. So
Mhm.
that is a thing.
Huge. Yeah,
So
refrigeration, that's the big part of the emissions is the refrigerated trucks for produce.
Mhm.
It's a
Yeah.
big deal.
Yeah, that's something that I was not aware of. This whole global food shipping, or you're talking about food lasting longer. Now I'm here in Denmark, which has plenty of farming, but not that much.
Yeah.
A lot of produce comes from elsewhere in Europe, and you
Or
have to
even
eat it the day
Africa.
you buy it. You have to
Yeah.
eat it the day you buy it, or you're gonna throw it away. It's that fast.
Yeah, so I mean, next time you go out to the grocery store too, look at the little tags on your fruit that you buy and see where they're from, because you might start to notice, okay, if I go to this store, they have a lot of food. In California, there's some stores you can go to where it's all from local farms, and there's some stores you can go to that like to wrap their vegetables in plastic, and they're often from Mexico, and so they have to travel a lot farther. And so, just it's something to educate yourself about so you can make good decisions.
Yeah. And yeah, just know that the carbon footprint of the more distantly traveled foods is going to be higher than something local,
Buy
more
local.
likely. Yeah. It's complex, though. It's a very complex system. But yeah, like just that the old saying, think globally, act locally, it just keeps coming back to it's a very accurate statement. All right, Blair, you had
Mhm.
one more story
Oh,
for this
I'm
first part of the
so
show.
excited. Are
I
you all ready? Everyone get snuggled down in your bed. Pull up
No.
your covers. Get your eye mask just right on your forehead, ready to come down. Put
No.
on your white noise machine, and I want to tell you about your face mites
Oh gosh.
that like to come out at night on your face.
It's
Or
nighttime, and the mites are
yeah.
crawling out of the pores in your skin and
Mhm.
crawling across your face so that they can mate with the other mites, and they're eating your dead skin cells. And oh,
Yes.
Demodex, we love the face
Oh,
mites.
face mites. They're
Face
passed on
mites.
during birth. Did you know that? They're
What?
carried by almost every human. They measure around 0.3 millimeters. They're found in
Tiny.
hair follicles, on your face and in the nipples, and on your eyelashes. That's why you're always supposed to wash off your face makeup. For those of you that wear eyelash makeup, you're supposed to wash it off before you go to bed because the face mites like to eat the makeup. Then they also mostly eat sebum, which I had to look up. Sebum,
Sebaceous secretions?
exactly. So sebum, as in sebaceous glands, as in sweat. It's oily, waxy substance produced by your sebaceous glands. And so these little buddies, these little co-pilots on your body that are always with you, somebody did some DNA sequencing on these little friends, and they found that they're actually changing. They're becoming so simplified organisms that they may soon become one with us.
Ooh.
Yes.
Are they
The
still gonna
mites
crawl around
move
on our skins?
in, but they
Mhm.
never move out.
Exactly. So this is the first ever genome sequencing study on D. folliculium, folliculorum. Excuse me, I missed an R in there. D. folliculorum.
Folliculi, follicula,
Yes, there you go.
folliculorulum.
So they found that their isolated existence on us and that we are passed down from birth means there's inbreeding going on with face mites. We're going to make them even more appetizing to talk about the fact that they're inbreeding. They're causing them to shed unnecessary genes. They're narrowing down their genome so much that they are transitioning from external parasites into internal symbionts. They found these mites have a different arrangement of body part genes to other similar species, which is most likely due to them adapting to the sheltered life inside our pores. We are giving them such a good life that they are throwing away unneeded genes. They found the following. They're extremely simple organisms with tiny legs powered just by 3 single cell muscles. 3 cell muscles for their legs. They lack UV protection, which is why they have to come out at night. And they have lost
Oh.
the gene that causes them to be awakened by daylight. why they're up at night. They are unable to produce
Oh,
melatonin.
wait. Say that again.
So they lack UV protection.
Yeah.
They have lost the gene that causes them to be awakened by daylight.
So is that I wonder if that's also helping so that we don't feel them crawling around when we wake up. Like they wait
We
till
wouldn't
we're
be able to
asleep
feel them. You
before
wouldn't be
they're
able-
like,
they're too
okay, humans asleep, let's party.
No. You wouldn't be able to feel them. They've also been left unable to produce melatonin, and which is how many small invertebrates are active at night. But they fuel their nocturnal activities by using the melatonin we secrete in our human skin at dusk. They're using us as the pharmacy.
Oh, we
They're-
give
I've
them
been
their wake
taking
up
extra-
power.
If
Yeah.
I've been taking extra melatonin lately, does that
You're
mean
feeding them.
Yes. You're making them have a disco on your face.
Oh,
Their
Boy, that's gonna help
reproductive
me sleep.
organs. Come on now. We got to talk about the mites' reproductive organs. They have moved interiorly. So the males have a penis that protrudes upwards from the front of their body, which means they have to position themselves under the female when mating. And they copulate. I want you all to picture this now while you're snuggled into bed, as they both cling on to the human hair. One of their genes has fully inverted, giving them a particular arrangement of mouth appendages which protrude extra for gathering food. That's just for fun. The lack of exposure to potential mates that could add new genes have set them on this course for what may also be an evolutionary dead end. We might lose our face mites.
No.
If we do, this is
I
actually, I
support
want to
my
talk about
face
this
mites.
for a second. Yes,
But yeah.
we don't know what will happen if we lose our face mites. Are they keeping something at bay by occupying our follicles? We don't know. Are
So
they
what I
cleaning us?
wonder, like, is there are they keeping down bacterial infections? Are they are people less likely or more likely to have acne if they do or don't have face mites? I want to know the answers to these questions. If
Yeah.
I am wearing sunscreen, what is that doing to my face mites? If I am going to the spa and having acid peels, what does that do to my face mites?
It's
I
a
want
great
to know.
question. Yeah, and speaking of healthy skin, apparently some people like to blame their face mites, not me, but some people like to blame them for skin inflammation because there is this theory that they don't have an anus, and so they actually just accumulate feces throughout their lifetimes, and then
Yeah.
they just kind of explode when they die.
Yeah, that's not true.
No, the DNA tells us that they do have an anus, and so they have been unfairly blamed for these
DNA
skin
says.
conditions. Thank you,
That's
DNA
awesome.
analysis.
Because
Yes.
that's like the one thing I remembered
mites have
about
an anus.
face mites is that they never went poop. They just ate until one day they just they burst.
It's
Yes.
not true.
Oh, well, that's good to know.
They're just
So
pooping
wait,
all over your
where's
face
it
all
going?
the time.
Ah,
Yeah.
that was
But
not
it's
really
just
better.
Tiny bits.
Not really
Tiny
better.
It's better.
I mean, perhaps they are leaving their frass. Would it be frass if they're a face mite?
I don't know.
I don't know. Maybe they're leaving them in the hair follicles, and that is leading to acne and inflammation. Maybe that's where they relieve themselves. I'm gonna guess they don't control it, and it's just kind of whenever. They don't seem to be that complex of an organism.
No.
No.
Yeah, they have a midden like a rhino.
Like,
this one pore.
This spot right over here.
Yeah.
That's my face mite midden.
Uh-huh. Exactly. Yeah. So anyway, face mites, we've done the DNA analysis. They are complicated, but getting less so. And I do hope that they don't go extinct. You hear that, buddies? Just here we go.
I'm gonna wash my face
Are
and
you
take
sure
off
you
my
can't feel
eyeliner,
them?
but I do hope that I do it in a nice way. Keep my face mites happy and healthy. I want a happy skin ecosystem. That's what I want.
I can feel my face mites now. But it's sort of like paying attention to your own breathing. Like now that you focus on it, and then now I have to control the breathing. This right now, I can feel the face mites.
Great. So I'm going to add to that by telling you I looked up what Kiki was kind of asking about. They are arthropods. They are, in fact, arachnids. So they're cousins of spiders and ticks. You're welcome. Enjoy. Teeny tiny
little
spiders on your face.
little tiny spiders that live in your face.
Or ticks, even worse.
Oh, they're mites. They're face mites. Oh,
Now I gotta
everyone.
find a kind of bug that I can put on my face that'll eat them.
No.
And then
Let them
I'll have to
be.
find another kind to put until I get one big enough that I can see and just. And then
I really do want to know so much more about face mites and their role in the face. And there are so many questions. What is it?
I'm
Derek
usually a
Schmidt is
curious
asking.
person. I don't want to know any more about it. I want to never hear about it again and forget that I learned
Well,
something.
that's too bad, Justin. If you're going to keep being on this show, I will bring them up again.
Like, is this why? I mean, washing your pillowcase. Are you washing away the dead face mites when you sleep on your stomach, on your face? Like, what happens to the face mites that are on the pillow side versus the not pillow side? I'm
do
so
they go-
curious.
Are they anywhere but the face? Do they just stay to the face?
Yeah,
Are they
I
like
said
whole
the
body?
nipples is the other place.
The nipple,
Yeah.
right?
Mhm.
But they're not like. Are they like also all on the arms and everywhere else? Because if they're everywhere, you're just kind of.
My understanding is they
do they
congregate. They congregate in certain areas.
Well, yeah, the face has the best view.
Yeah. It's-
Hey, everybody, I'm taking a ride. It's like riding on the top of the Statue of Liberty as she walks across New York City.
Now I feel like a giant spaceship.
Yeah,
You
kind
are.
of
you are.
animatronic robot. Kind of like that.
Spaceship human. Everyone, might you enjoy This Week in Science? We hope that you're enjoying the show this week. This is This Week in Science. If you are enjoying it, please take a moment to subscribe. Head over to twis.org and click on the subscription links on our page there, or head to your favorite podcast provider and look for This Week in Science. All right, we're going to come back to a wonderful part of the show that we love. Not the COVID part of the show. Oh, no. We're skipping that one today because, everybody's skipping it these days, even though everybody's getting it. But anyway, it's time for Blair's Animal Corner.
With Blair.
Yeah. Oh, hold on, because, I have to do the thing. Wait, I'm going to make you go backwards now. Say it again.
With Blair.
She loves our creatures, great and small. Biped, millipede, no pet at all. If you want to hear about animals, she's your girl. Except for giant pandas and squirrels.
What you got, Blair?
Oh my gosh, I am so honored to bring you tonight the story of teeny tiny frogs that are bad at jumping. So, Kiki, I'm going to need you to share some video. It's in the second link.
Oh, okay. I'll get in there.
And this is a story about Brachycephalus, which is a genus of frogs. They are called pumpkin toadlets, some of them, and some of them are called flea toads. They have become so small that they're no longer able to maintain balance during quick maneuvers like jumping. They're also just really bad at walking in general.
Oh, they'll all be dead in 10 years.
That's right.
Maybe.
They have no balance.
Maybe. There's a reason evolution has done this, which is part of what we're going to talk about. But so they lose control of their trajectory mid-leap. It starts like it looks like it's going okay, and then it just starts to go bad. Here, we're going to watch a video. And so they're kind of puffing a little straw
No. Oh,
at these
no.
frogs, and it causes them to jump. And for almost half of it, you're like, "Oh yeah, you got it." And then things just go terribly wrong. I can't.
So is
even...
this?
I need
That's
to play
really
that over
bad.
again.
Is this a hybrid by any chance?
No. So this is a whole genus. There's a bunch of species. You're going to see a bunch of different species in these videos.
Okay.
And so they're all just terrible at jumping.
Oh, gosh.
It's like they freeze as
Yes,
if they
exactly.
they leap and then get to the full extension with their legs straight and their arms out. Their body is extended and then they get stuck.
Mhm.
And then they just tumble head over tail until they land however they land. That's
It's,
gotta
yeah,
be
it's like
good
if
for
a
these little
cat fell
dudes.
off of a dresser, but instead of like contorting to land, it just stiffened. And
Yes, exactly. Yeah. And so I guess the idea is they really don't try to jump as a defense mechanism. It's a last resort for them. And so if they're actually jumping, they're just like,
It's
I
the
hope this
freeze-
works out. And so they're just going, they freeze up, they're going for it.
No,
And-
it's the freeze game. They got a buddy you can't see off camera there, who as
Yeah.
soon as he jumps, they go freeze, and he's like,
And they land on their legs and their legs don't bend and their
Yeah,
balance is all off. They're
completely stiff.
they're like it's like a not fight or flight, it's the freeze response.
Exactly. There should be 4 videos. So there's some other ones.
I'll go find them.
Of some other just poor
gosh.
examples of
So
jumping.
there's like, this isn't just this one frog. It's not a hybrid, because I kept thinking it's got to be a
No.
hybrid. It's like one
No.
frog that had a big upper body, one frog that had different legs, and then they got a hybrid of the two, and now it just jumps wrong.
Yes,
because
here's
it's-
another one. Oh no. Oh!
How does it even exist? How did it get
So
this
that's what I'm
far?
going to tell you.
That's what I'm
So,
curious about.
Yes. So the reason they exist,
I was hoping.
I'm
you'd
so
tell me.
sorry. If you are listening to this as a podcast,
You need to
You
look
haven't
for
heard
these
it
frog
anyway.
videos.
if you're driving, pull over and look this up right now. Go to the show notes, go to twist.org, look at the latest broadcast, click on the link, look at these videos because
Did you say the latest frogcast?
yes, the latest broadcast. If I didn't, I wish I did. But okay, so the reason they are so bad at jumping is that they are small. They are so small. They have gotten so small that they are no longer able to maintain balance. How does that work? They looked at their vestibular system. This is a 4-year initiative among 18 institutions. They looked at 3D models from CT scans of more than 20,000 museum specimens of this genus across the United States. They stitched together all these different frog scans to get as close as they could to a comprehensive model of their vestibular system. The vestibular system is a fluid-filled network of spiraling chambers inside the inner ears. It looks kind of like a nautilus. And when it moves, when we move around, the liquid moves along with it because, gravity. And that brushes against these tiny hairs that send electrical impulses to the brain that tell us where our orientation is in space and how to maintain balance. So kind of related to the whole one-legged conversation we were having earlier. Sometimes people's vestibular system starts to go bad as they get older, and that can have to do with their balance issue. So anyway, the vestibular system is the main mechanism how most vertebrates are able to do the acrobatic things that they do. What scientists have discovered is that there is a specific restraint on the size of a vestibular system to allow function. For example, baleen whales have vestibular systems. Their chambers are only slightly bigger than ours, even though they're huge compared to us.
Huge.
They don't need to be that
It
much
gets the
bigger.
job
They
done.
might not function if they get too big. Similarly, the chambers within the skulls of these toadlets are as large as they can possibly be in that small head. They look like overinflated balloons on these scans. So they are as big as they can possibly be relative to their heads. But even in that case, it's not big enough. The vestibular system does not function enough for them to maintain balance when they jump.
Because they dwarfismed down to the point where now I get it. Okay. So there was a bigger version of this frog, did just fine throughout evolved, could do the thing. And then, I don't know, they got isolated on an island or however it happens. Dwarfism took over.
So
They shrunk.
they don't really think this is island dwarfism. They think this is actually specifically an evolutionary tactic, and it is because they are so small, they go unnoticed. And so the benefit of being small outweighs the benefit of a speedy getaway. So basically, they're more likely to just go unnoticed than have to run away from predators, and therefore it is more important to be small than to be graceful in this case. So I will also throw out there. Yeah, go ahead.
I was just going to say, and that it's just a huge evolutionary benefit. The size has allowed them to survive, whereas their vestibular system would not. If they were continuing to be predated upon heavily, then they would have been eaten because they wouldn't be able to jump to get away.
Right.
So yeah, so this makes sense. Yeah. So it's really just the vestibular bones and the whole system's gotten so small, there's just no space for anything inside of it.
Right.
It's just a little thing. It's a little nubbin that doesn't work it doesn't do nubbin.
Yes, and MT in the chat room in YouTube brings up a really good point. They still have a jump reflex or instinct. Yes. Yeah. So they still have this instinct to jump to get away. That is still their best case. If they've been spotted, they can take a tumble and maybe kind of right themselves and hide again, and they'll do okay. And so who knows? Maybe eventually, if they keep getting smaller, they won't even be able to jump anymore. I don't know. But just to add to the craziness of these frogs, also just FYI, they can't swim. They don't have tadpoles. They don't get around much. When they've monitored acoustic behavior of them, they record the same individual at the same spot over the course of a year. They really don't move around. They find a good hiding place. They stay there. And in 2019, I do believe we reported on it on this show, they were found to have fluorescent bones.
Whoa!
Oh, the same guys. Okay.
Yeah. Fluorescent bones.
Yeah. So these guys are just a barrel of fun. Yeah.
They're fluorescent bones, but I can't jump.
No.
So anyway, and they don't have tadpoles. I thought that was just kind of an obligate frog thing.
No, there are a few kinds of frogs that will go straight to froglet.
Wow. Okay.
Yeah.
Probably smaller, more likely to go straight to froglet, but yeah, fascinating.
Yep. Yeah. So there's the awkward frogs that are still winning at life by being very, very small. And now from frogs to frog-eating bats.
Woo, let's go.
Yes. I want to tell you about researchers who train bats to associate a ringtone with food. What they found was the bats remembered the ringtone for up to 4 years after having been released in the wild. This is, as far as researchers know, the first long-term memory study involving animals being released into the wild, recollected, and then tested again. So there's been lots of studies on long-term memory, sea lions for 10 years, tortoises for 9 years, dolphins for 20 years, but those were all animals that lived in a lab or in captivity the entire time. So this was an important study because they taught them this association, released them, and then brought them back. And they used something to teach them on that they didn't interact with, most likely, in the wild for the entire time they were away from it. So they really did remember this kind of in isolation. So the way that they did that is they took frog-eating bats from the wild. They did a series of cognition tests where they were exposed to an attractive sound, which in this case was the mating call of the male Tungara frog, which is one of the things they love to eat. They flew to that sound and they were rewarded with a piece of food. And then over time, they mingled that sound and gradually replaced it by a ringtone. But they still gave them a reward. So then by the end of the study, they were able to introduce other ringtones, but they specified this ringtone means food, and the bats responded, and they were able to discern the differences in the ringtone. Kiki, you're muted. Oh yeah,
that would get annoying after a while.
Yes, although I
Poor
do wonder
bats.
if any kind of lost researcher wandered out into the forest and they started getting text messages, "Where are you? We can't find you." And then they just started getting hit in the face by bats. Where's my food? So once they learned the ringtone, they were microchipped and returned to the wild. Then they collected them at a year, 2 years, 3 years, 4 years, and they were able to test them on their retention of that ringtone. And so it was successful. They found that 4 years, even though that's a long time to hold on to information you might never use again, 4 years was enough for them to retain that useful information in that one instance and be able to associate it with food. So this is beneficial for a bunch of reasons. Obviously, it has an impact on wildlife conservation and reintroductions to the wild. It's a reminder that whatever you kind of teach them when they're in captivity and what they learn to associate, that could carry on with them for a very long time. But it could also impact future studies. This is a really cool proof of kind of the mechanism that we could use with other animals in the future. And it shows that it's possible. If these bats were able to retain these memories for this many years, then that is a motivation to try it with other species as well.
Yeah, I mean, this is the big question for longer lived species. How long do they retain the information? We have this long held assumption that, oh no, they're not going to remember any of this stuff, or we just, haven't really tested these things.
Yeah.
Yeah, and those number of years that those different creatures, the sea lion 10, the dolphin 20, those like they tried it 11 years and it's like, sea lion forgot. Or is it that the study was, they tested out to 10 years and it could go beyond that?
Yeah, see, that's the question. Yeah.
Right. And it depends on the study. Some studies, they do find kind of the threshold, and some studies just end.
Right.
And so
we
it's
did it 20 years
at
apart.
least as long as, I mean,
Yeah.
in this case, 4 years, it's a suspiciously similar number to how long it takes to get a PhD.
Yeah, it is. Yeah.
Suspicious.
Anyway, something
We'll
to
be
consider.
expecting to see another paper out of this in another 4 years or so after another student picks up the project.
Yes. Or the main
Longer.
researcher carries it on to their postdoc. Who knows?
Exactly.
Yes, so animal memory, another episode of animals are smarter than we gave them credit for.
It's an amazing long memory. I mean, long memory. It's good if you're long-lived to have a long memory. That's going to help with survival, and it'll because it makes things easier. If you remember things, then you can discriminate between choices that need to be made. Yes. All right, Justin.
Uh-oh.
It's time for you to tell us about some
Let's see.
science.
What we got here? So, what is it? It was about 100 years ago. On the suburban outskirts of Canterbury, Kent, in jolly old England there. Workers unearthed some odd-looking artifacts, hand axes and other odd tools that they found while digging ditches back in the 20s. These artifacts were determined to be very old. So they sent them to a museum where they have been stored ever since.
Yeah, it belongs in a museum. I get it.
It looks very old. So you give it to the museum folks because that's what they handle all the very old stuff. Now researchers have gotten around to asking just how old are they? They dated the artifacts using infrared radiofluorescence, IR/RF dating, a technique which determines the point at which sands of grain were last exposed to sunlight. So they went and they re-excavated the site, and they found more artifacts, and actually some different artifacts, scraping tools and some other things that they hadn't originally found. And they have dated this to between 560 and 620,000 years ago. This is quoting Dr. Tobias Lauer from the University Tübingen in Germany. Tübingen? I don't know how to speak German. This Dr. Tobias Lauer, who did the dating of the site. This is one of the wonderful things about this site in Kent. The artifacts are precisely where the ancient river placed them, meaning we can say with confidence that they were made before the river moved to a different area of the valley. So these are artifacts in situ. They were in a river. They got tumbled along by an ancient river. That ancient river isn't there anymore, so they at least can, get some of their dating that way. That gives you an older than date to start with. So you have also something that you can help verify or compare to your instrument data, your IR/RF in this case. So you also have something that another data point or another way of looking at that. So anyway, this is Dr. Alistair Keeve, University of Cambridge, directed the excavation. The diversity of tools is fantastic. In the 1920s, the site produced some of the earliest hand axes ever discovered in Britain. Now, for the first time, we have found rare evidence of scraping and piercing implements at this very early age. I think this is Homo heidelbergensis, a hunter-gatherer who ate very diverse things. With the scrapers and these pokey things look like they may have been for processing of animal carcasses that they think could have been used for clothing or even for housing. So, based on the number of tools and the type of activities that they estimate were taking place, it looks like life was good 600-ish thousand years ago in England. Now, this is before England's first Brexit. This is while England is still, I think, attached to the rest of Europe. There isn't water separating, so they didn't have to sail or swim across. They could have just walked there. Dr. Thomas Profit from the Max Planck Institute of Evolutionary Anthropology analyzed the artifact, said scrapers during the Paleolithic are often associated with animal hide preparation. Finding these artifacts may therefore suggest that people during this time were preparing animal hides, possibly for clothing or shelter. Yeah. So it is possible the European populations of Homo heidelbergensis evolved into Neanderthals, while a separate population of Homo heidelbergensis in Africa evolved into Homo sapiens. That is one of the theories. Well, really old, though, this is still older in England. They have a collection of footprints in Happisburgh, Norfolk, that date from 840 to 950,000 years ago. So things have been walking around there for quite some time.
Fascinating. Yeah. I mean, we always have thought about there's Homo sapiens and out of Africa, and okay, then there's Neanderthals, and okay, we were separated. Oh, wait, no, we were living in the same places around the same. Oh, what are these other species? And now you're kind of off the cuff comment here. Oh, maybe hypothesizing that Heidelbergensis could have become Neanderthal. What? Like this? I'm like, what? This is awesome.
Yeah, it's all everything and it's all nothing.
Yes.
It's a soup. It's a stew. It's human stew.
Yeah.
How fantastic!
And that's why I'm skeptical that of that idea, the Homo heidelbergensis just becoming Neanderthal, and then the every information that we've gotten without fossils. Once we get a fossil, we learn a completely different story. That's the one thing they don't have here. This is stones that got maybe they were washing the axe and it ended up down the river.
Yeah.
These are lost tools into an ancient river. There's no skeletal remains anywhere. There's no jawbone or tooth or anything to tell us who
it was.
We can speculate.
Who are you?
We can have an educated speculation. And one thing we've learned a lot about these educated speculations is it's weirder, man. It's always weirder than
It's weirder than you think it is. Yeah.
The history of mankind is weirder than we ever thought it was. And there's a new therapy to combat cancer. Could one day cure it. Actually, it's curing it already
Wait, what?
in
Say
mice.
more.
Ah,
In mice.
But-
in mice.
But this is another colon cancer cure. We're finding multiples of these now. I feel like these are just, nobody should die of these things. This is coming soon. A combination of a plant virus and an antibody that activates the immune system's natural killer cells is showing tremendous early results in mice, according to researchers at the University of California, San Diego. In mouse models of colon cancer, the combination therapy eliminated all tumors and prevented their reoccurrence when they tried to give the mice cancer again. And the immune system had learned well enough to still be on the lookout and attack the cancer cells. So their results, 100% survival.
Of the mice.
Yeah.
100%.
For
Amazing.
infinity. They're still alive today.
They are not, probably.
No,
never
you're
mind.
still alive.
They-
We
it's
must
a-
kill
it's-
you
it's
to find out why.
immortality. Yeah.
Well, they survived the experiment, and
Yeah.
then immediately after, they were probably incinerated.
Yeah.
All right,
Yeah.
so put that in your colonoscopy pipe and smoke it, because the disease is soon likely to be a thing of the past. Therapy also increased survival in mouse models of melanoma. The work is reported in a paper. Reported in a paper published in Nanoletters. Fun fact: Nanoletters is the only scientific journal which requires magnification to read, even if you have perfect vision.
Ha-ha.
Really? So they tried it also with just this plant virus, and it had a positive effect, but it wasn't nearly as good as when they combined it with this antibody. So there's by having this, and this is the Cowpea Mosaic virus, which is a plant virus which normally affects legumes, but is completely harmless to animals and humans. And the antibody is anti-4-1BB, which you probably never heard of. So the antibody has the ability to, well, between the two of them, they can actually bind. Okay, the whole problem with the natural killer cells, they're there. They're around the cancer. They find the cancer. They say, hey, there's cancer here. And they create an antibody by attacking it. And that antibody is supposed to go out and train the rest of the cells. But there's not enough of them. There's not a big enough concentration of them that the immune system, because the cancer is horribly, terribly afflicting of an illness as it is, it's not actually proliferating that many cells at any given time. It's not, you're not going to get the antibodies up to the level where you're training the immune system to go and kill it. So,
Right.
And they can also bind. They also have a ability to bind some of those antibodies when they get discovered to the cancer cell, so that it doesn't even get out into the mix and get trained. Something between this virus and this antibody, they're able to invite. I guess the plant virus attracts other natural killer cells, and the antibody keeps it from sticking, so that the other antibody keep this sticking mechanism of the cancer at bay. So it gets out. It gets the message out, is basically what happens. And what was also cool is that the immune system, once trained, had immunity memory. So they went back and tried to, after the mice, they gave them the cancer and the melanoma, and then the mice survived. They recovered. And they said, let's give it to them again. Oh, and they recovered again.
It's amazing. So once you prime the immune system to recognize the cancerous cells, then it's ready to do it. It's like, I can protect myself now.
It's almost like an inoculation.
Yeah, that's kind of
Ooh.
his thing. It's like, I just
Yeah.
go ahead and give everybody this thing now and train up the system.
Might be a little early, but yeah, that's a fascinating.
Well, you'd
Okay,
have to give everybody cancer.
if this can move quickly through the pipeline.
That would be pretty
would
wild if, as a kid, like now you get your Tdap or whatever. Like like as a kid in the future, you could get your anti-cancer series of shots as a
Well,
child
there's
and
one
never have to
already.
worry about it again.
HPV vaccine.
Yeah,
I mean,
you're right.
we don't call it a vaccine against a sexually transmitted disease. Call it a pre-cancer treatment.
Cancer
So it's
Totally.
vaccinating yourself against cancer is what you're doing.
Yeah.
Yes. Oh, this is amazing. Oh, the immune system is so interesting. Now I'm just like, this is such cancer ecosystems, the immune systems, they all work together so in such a fascinating way. Let's talk about brains and things.
Yes.
What shall we discuss now? I would like to talk about, let's see, what story did I bring in here? Oh, I think I put the wrong link in my rundown. That's too bad. What I wanted to talk about is how your breath can be used to identify you before we start talking about some other things. Yes. What does your breath say about you? Well, a new
But I love
study.
garlic. Oh, sorry.
Yeah, exactly.
Mine says coffee and cookies this morning.
Well, this new study out of Japan, they have been looking into ways that biometrics can be used to identify individuals. So we all know fingerprints. We know pictures of your pupil, that your pupil is like a fingerprint, even more unique than your fingerprints, in fact. People use teeth. They use, they're starting to look at microbes. They've been starting to also look at the chemical compounds that are released from your sweat. And so these researchers said, wow, we can take a look at stuff that comes out of people's sweat. But these compounds that are unique to individuals are usually found in parts per billion. So they're really, really tiny amounts. And so they're hard to find. And they said, what's another thing that we could possibly use? And they went, oh, I know. And so they started measuring compounds that come out in people's breath. And, you might think, oh, all the stinky food and all the other things. They found 22 compounds that can be used to identify people. And so they stuck these compounds on a chip and they started testing them on people. And so far, it's a very small pilot study. They've been looking at like 20, 30 people. It hasn't gotten to the mass testing that it needs to be to really be useful. But at this point, as they have been developing this fingerprinting system based on breath, it has been over 97% accurate in identifying individuals. That said, all the individuals who were involved in this study had to fast for 6 hours before the study, before breathing on anything.
Okay, before you enter the safety room,
you can't have had anything to drink. You can't have had anything to eat. You can't.
I'm never getting in.
No, to get into the safe or to get into this like panic room or whatever, you can't get in. Don't eat for 6 hours, then come back.
I would like to get into my safe deposit box, please. Yeah. Go fast for 6 hours and come back later.
Is this all the microbiome? Is that the idea?
Not necessarily. These are volatile compounds, chemicals that are released from, it's in part due to the microbes that are probably active in our systems. But this is also, because it's stuff coming from, it's your lungs, your airways, your mouth, your nasal system, so they're probably going to be bacterial components that create the uniqueness to individuals. But
they probably could do this. They probably could do this with the microbiome, but it wouldn't be a breath analyzer.
No.
Yeah.
You'd have to have a very specialized diet for that one too, I think.
You would.
Uh-huh.
Right. But anyway, maybe one day, your smartphone with a little breathalyzer thing, maybe instead of your thumbprint or a pupil scan to get into your bank app on your phone, you blow on your phone and your phone goes, "Oh, it's you. Here's all your money."
And it still won't work because we'll still be wearing masks.
Very
That's
possibly.
hilarious.
Very possibly. And my final story for the night is a little bit itchy. Itchy. I mean, you think about those
Oh
face
no,
mites.
this is like yawning.
Why are
Mhm.
you
Talk about
giving me
being
the face
itchy.
mite story?
When you
again?
think
Oh,
about those
gosh.
face mites crawling
I
on
don't care
your
about that. I just
skin, like
No, I can
maybe
feel them whenever you say these things.
maybe
I
they
can
make you want
suddenly.
to scratch.
Why
Maybe
can I
they
feel
start a
them?
little sensation of itch, and they make you want
Yeah.
to
Oh,
scratch that
oh.
itch. How does our body understand that physical itch sensation that can be caused by a loose strand of hair or a small parasite living on the surface of our skin? A study published in Nature this week has discovered that is a subset of neurons that was previously thought only sensitive to chemical stimuli for inducing itch, but they discovered at the Scripps Research Institute that there are proteins, ion channels, for which the Nobel Prize was won recently. The piezo 1 and piezo 2 proteins, these ion channels, very important to sensations of touch. Anyway, they've discovered that these chemical stimulated itch sensors, these neurons in our skin, they also have these ion channels that allow to be stimulated by physical touch. And they never knew that before. So it's a researcher working in the Nobel Prize winner's lab, looking at the mechanisms of touches that lead to itching and scratching. It's really a misunderstood or understood area of sensory biology.
Hmm.
And she put a bunch of these neurons in a dish and didn't know what was really going to come out, was staining them for the piezo ion channels as a control because they were not expected to be there. But then they found them. So the control ended up being the study. Ended up being something they did not expect whatsoever. And they found that these chemical stimuli responsive itch sensing neurons express the physical ion channels. And so it's a whole new pathway. And so they genetically modified mice to not have the piezo ion channel in their itch sensing neurons. And guess what? They didn't itch and scratch so much. And then when they gave the piezo 1 back to the mice, then they started itching and scratching more. And so this is a new understanding of these itch receptive neurons, that they have an ion channel that we didn't think they did before. So
Did those did they cough as much? Because I was just thinking about how you get an itch in your throat, and it's a very similar sensation, but it has this kind of compulsion where then you have to cough.
yeah, well,
I
they weren't
wonder if it's
looking.
similar.
Yeah, they weren't looking at coughing in mice.
Yeah, of
So
course
I
not.
have no idea. But they were looking at this in skin, but since the
Right.
epidermis, it's the inner lining of the airways is still kind of epidermal. It very well could be that physical. I mean, and the inner
So
lining of
that's
the airway
study I
does
want
have
next.
little hair growth in it. Anyway, yes, we'll get rid of the cough. But while
yeah.
we understand how this touch sensitive neurons, how these neurons are sensitive to touch and chemical stimuli, this could lead to additional ways to treat dermatitis or eczema or other physical itchings, things that get
Chicken pox.
stimulated, disorders that lead to itching over time, itching and scratching significantly, which can really harm your epidermis. right? Can harm your skin, which is the barrier between you and the environment. So it's very important. And apparently 10% of people will suffer from chronic itch at some point in their lives. It's a lot of people who are just itching.
Yeah, you know who
wanting to
those
scratch.
people are? You know who those
Yeah.
people are? Those are people who learn about face mites.
I mean, some of them, but not all of them.
Maybe you'll be scratching your face as you fall asleep tonight, Justin.
Yeah, I wonder why that is.
I'm gonna say
Instead,
goodnight.
he's gonna be
each
smacking
one,
himself in the face.
one by one. Good night. Good night. Good night.
And I kiss you goodnight. And I kiss you goodnight. Good
And
night.
I'm
Good
just
morning,
gonna
little face mite.
don't mind me. You all sleep tight. I'm just gonna put myself under this UV lamp.
There was speaking about that, older men, not in their 50s, 40s, 50s, but in their 60s, older men in their 60s exposed to UV light while they were sleeping had worse sleep and more health problems than men than women or younger men. So there's something that happens as you get older, being exposed to UV light while you're sleeping that's bad. So don't sleep with the UV lights on as you age.
Blue light is worse than any of them, and they can cause cancers and everything else because they mess with your melatonin levels or something in their I don't know.
Your face mites won't like that. You can't mess with your melatonin.
The best actual way to sleep is in darkness. Light therapy.
Yes.
Scam. Don't fall for it.
I love darkness. Hello, darkness, my old friend.
Have we
Bringing
gotten
face
to the
mites
end?
here again. Dun dun dun. It's time. We've hit the end of the show. I did it. We did it.
We did it.
We did it. We did it. We did it. Thank you so much, everyone, for joining us, bringing your face mites to the show. I hope they learned as much as we all did, and I hope that your little spaceship face, that it brought a little bit of entertainment to those face mites and to you. Thank you for joining us for sure. I want to say thank you also. Shout outs to Fada. Thank you so much for your help on show notes, show descriptions, and social media. Thank you, Identity4, for recording the show. Gord, Arnlor, others who help keep the chat rooms a nice place to be. And Rachel, thank you for editing the show. All of you. I couldn't do the show without you. Hosts, co-hosts, thank you. It's amazing. And thank you, TWIS patrons.
Yes.
Thank you to Teresa Smith, James Schofer, Richard Badge, Kent Northcote, Rick Loveman, Pierre Velazar, Ralphie Figueroa, John Ratnaswamy, Carl Kornfeld, Karen Tazi, Woody MS, Chris Wozniak, Dave Bunn, Vegard Shefstad, Hal Snyder, Donathan Stiles, aka Don Stylo, John Lee, Ali Coffin, Gaurav Sharma, Ragan, Derek Schmidt, Don Mundis, Stephen Alberon, Daryl Myshak, Stu Pollack, Andrew Swanson, Fredis 104, Sky Luke, Paul Ronovich, Kevin Reardon, Noodles, Jack, Brian Carrington, Matt Bass, Vote Beto for Texas, John McKee, Greg Riley, Mark Hessen, Gene Tellier, Steve Leisman, aka Zima, Ken Hayes, Howard Tan, Christopher Rappin, Dana Pearson, Richard, Brendan Minish, Johnny Gridley, Remy Day, Flying Out, Christopher Dreyer, Artyom, Greg Briggs, John Atwood, Rudy Garcia, Dave Wilkinson, John Rodney Lewis, Paul, Philip Shane, Kurt Larson, Sue Oster, Jason Olds, Dave Neighbor, Eric Knapp, EO, Kevin Parachan, Aaron Lutzen, Steve DeBell, Bob Calder, Marjorie, Paul Disney, David Simmerly, Patrick Pecoraro, Tony Steele, and Jason Roberts. Thank you for all of your support on Patreon.
A lovelier bunch of people there have never been.
Oh, what a lovely group of people. And you, if you're interested in being a part of that lovely group of people, you can find a link to support us on Patreon at TWIS.org. On next week's show,
we will be back Wednesday, 8 p.m. Pacific time, broadcasting live from our YouTube and Facebook channels and from TWIS.org/live.
Do you want to listen to us as a podcast? Maybe as you feverishly scrub your face before you go to bed? Just search for This Week in Science wherever podcasts are found. If you enjoyed the show, get your friends to subscribe as well.
For more information on anything you've heard here today, show notes and links to the stories and studies will be available on our website, www.twis.org. And you can also sign up for a newsletter that won't have anything to do with face mites.
I can't guarantee that. Yeah, we'll see. You can
Oh,
contact
it's
us
gonna be a
directly.
special
Email
edition.
Kirsten at kirsten@thisweekinscience.com, Justin at twistminion@gmail.com, or me, Blair, at blairbaz@twist.org. Just be sure to put TWIS, TWIS, in the subject line, or your email will be spam filtered into our junk inbox, and it will land as gracefully as a pumpkin toadlet. There.
Hey,
Which is to say, not very gracefully
No.
at all.
No. It'll fall right
Yeah.
off the screen.
You can hit us up for comments or story suggestions on our Twitter, where we are at Twist Science, at Dr. Kiki, at Jacksonfly, and complaints about the face mite story should all go to at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes to you in the night, please let us know.
We'll be back here next week, and we hope you'll join us again for more great science news.
And if you've learned anything from the show, remember,
It's all in your head.
This week in science. This week in science. This week in science. This week in science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth, and I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer, and it shouldn't be news, that what I say may not represent your views, but I've done the calculations and
I got a plan. I did the calculations. I got a plan.
Is it that time of the show when I get to sit down?
Yeah,
That's
it is
fun.
the Blair Sits Down show. Take a load off, Blair. This week in science. This week in science. Zazzle and Patreon for the support. Oh yes, Eric Knapp. Midnight Sun. Happy solstice, everyone. We are just past that most brightest day of the year in the northern hemisphere. Yes, summer solstice for us northerners.
It was 102 degrees yesterday at my work.
What?
Yeah.
In the Bay Area?
Yeah. I mean, San Jose. Gets pretty hot down there.
All right, Laura, I don't know Disturbed, I think. Should I look it up? I might like it. I'll look it up. I like lots of things. Yes, Blair was standing on one leg the entire
I alternate.
show.
I do actually spend most of the show on one leg. It's so my legs don't fall asleep.
Yeah.
So
Well, what you have to do is you have to, you not just, you have one knee and you hip to hip, leg to leg, foot to foot. You move it around. I'm very walky and movie, even though I don't know if you can notice it from the other side. On the below, I'm very mobile.
You're doing river dance. It's all
am,
below the belt.
I'm constantly stretching my legs too, stretching my feet and my ankles. There's constant movement over here. A metal band. Oh, I bet that would be great. I have to check it out, Arnlor. Put a link in the music section. Yes. Oh, yeah, Noodles. Now the 25% off code. I don't know why Zazzle decides things are going to be 25% off or not.
Zazzle is fickle. I've noticed. It's
I have
just
nothing to do with it.
I feel like the only times I've ever ordered from Zazzle are the day before a major sale. Always.
Right? like, oh, I'm gonna order something. Hey, wait a minute.
Mhm. Yeah.
Yes.
Should we watch more videos of frogs? I have more.
Do you have more videos of frogs
Of course I do.
bailing? This is, I mean, I feel like we need some heroic music, like Chariots of Fire, with these frogs. Oh my gosh.
I need to here. There, is that big?
Yes, it's big. It's
Okay,
big.
great.
Oh, Brachycephalus pernix. Brachycephalus.
Now also imagine they're just being blown on by a little straw. Oh.
I love that you're watching this again. I'm so happy.
I may have watched it 5 times last night.
I mean,
And just watch this one, the little leg. He's just like, all right, let me just, I'm gonna start to try.
and
to,
I
okay.
can roll over.
I'm gonna
I
roll
can
over.
do
I
it.
can do it.
Oh no!
I can-
Oh, that didn't feel so good.
Oh gosh.
Here we go. I'm ready. I can do this. Ready, set, go! Oh no! No! Oh.
Poor buddies.
Flew like a brick.
Somebody
Look at
needs
all of
to
these
like.
I love it.
frogs.
Somebody needs to edit in like a little mattress. Like, like make
So
it a
it's
mattress
not
where it's
just
like after a
landing on a
long
hard
day
surface.
of being a frog, and he's
It's
just like
hard being a
leaping
frog.
onto his back onto the mattress. Dun, 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 it's Frogman, able to leap tall buildings. No, wrong kind of frog. Unfortunately, sad fact. I got a superpower of a frog, and it turns out it's that one. It's bad at jumping.
I could just watch frog videos all day.
More frogs. More frogs.
Here,
Okay.
hold on. I'm gonna share another thing.
Okay.
I think I already did this, so it's a repeat, but we're gonna do it again. Just enjoy. This is the desert rain frog.
Night terrors. His eyes.
See him? You think he's gonna get taken down by that scorpion?
No, I don't.
I did.
Oh, can we not?
No, YouTube's gonna get us.
Oh no. Okay, I'll kill it.
Well, it's
I thought that was
you
just
can do
sound.
Reaction.
I didn't
video
know
or
it was
something.
video also.
I thought you could do like a reaction video, and we're doing it in the context of a sciencey show.
Okay. Well, anyway,
and the
everyone, I
context-
know
YouTube
desert
doesn't
rain
care about
frog.
context.
In
And
the
they,
context of a sciencey show, I would also point out that it had narrow slit, vertical narrow slit eyes.
Mhm.
Why is that, Miss Blair?
I don't know.
So
I don't know.
So it's because they don't have the muscle structure to like our pupils. They dilate. The whole it gets a big circle, small circle. A lot of animals don't have that same ability. So what they do is they just it's the same idea. You're making a smaller aperture, but they do it from across. It's pulling the ends as opposed to goats have that too. You ever see goats
Yeah.
that got the sideways? But theirs is
Yeah,
different
but theirs
because
is the opposite way.
right, and the reason for that is it's whether you spend your time looking down or up and down or to the sides. Which one matters to you more for your night vision, or for when you're adjusting for sunlight?
Okay, so what I just
Or
found,
moonlight.
I
Is
haven't,
it
Or
this
bad?
is something I used to have in my brain. Speaking of long-term memory, it's probably been more than 4 years since I talked about it, and I don't remember.
Yeah.
But so a very short dive shows me that goats, it's rectangular because they're awake during day and night. But foxes, cats, other animals with the vertical slits is because they're primary nocturnal. So they need to reduce blur from trying to see at
Mhm.
night.
That makes sense. Yeah, because goats, they are that wider, more rectangular. It's like an in between of the round pupil and the vertical slit. Fascinating. Except for when the cat is agitated and its eyes are completely black.
But I think it's also because there's a side to side because it allows, because it affects the vision if they're still looking out for predators versus the other ones are hunters. And so they're looking to track something as it's leaping away from them. I think it has something to do with that.
So at first when you put up this picture, Kiki, I thought someone got a TWIS wrap on their car.
That would be awesome. What happened to your car? I got a TWIS wrap, you know. A TWIS towel. Noodles got a towel.
Going to the beach
Yeah.
with that
Good-looking towel.
towel.
Gotta be using that towel. It looks great.
I need to
It's a
get that.
it's a fantastic towel, I have to say. I quite enjoy it.
Does,
I hope
you're
you
right. It does
enjoy
look
it
like
as
a
well,
vehicle.
Noodles. Thank you for purchasing a towel. I love this picture. Yay!
Oh yeah, and the octopus has a weird eye too.
The octopus
Oh yeah. Well,
does
I'd
have a
assume that
weird
has to do with
eye.
the fact that they
Yes.
see things that we can't see.
Oh.
And they communicate with colors.
And they're underwater. That's got to have something
Yeah.
to do with it. There's this
So
sideways
the pupil of
eye.
an octopus is circular in darkness. In bright light, it constricts to a horizontal slit.
But then there's like this woogly dude. He's like,
Yeah.
mine's gonna be a wave shape.
Well, that's a cuttlefish. Maybe it's
Oh,
cuttlefish.
because cuttlefish, they're not normal.
Cuttlefish. I have a cuttlefish. Modified horizontal slit
Another one.
pupil with a distinctive
Cuttlefish.
W shape. 2 suggestions have been made for the function. We don't know, ultimately. 1, camouflaging the eye. That would make a lot of sense. 2,
Mhm.
providing distance information.
Oh, interesting. Because of parallax, even within the eye, probably, maybe.
Balance out the uneven light levels in their habitat. I mean, they do live in the waters where there's like pillars of light and then darkness, and it's all, the waves, and it's chaos.
It's underwater. It's chaos.
Chaos.
Absolute chaos. Right? Sometimes you don't really think. It's like, oh, it's just what it is. But then someone asks a question. Why is it that shape? These are very good questions.
Somebody said squid eyes are the weird ones. I feel like squid eyes look pretty normal. I think nautilus eyes are the pinhole eyes. Those are bizarre.
Ooh.
Yeah.
That is
Pinhole pupil.
relatively bizarre. So it's like a pinhole camera.
Yeah, it has no lens.
Well, I mean, it is an old. The nautilus is an old creature, right? It's been around for a while, so a simplistic pinhole eye with no lens would be a pretty easy eye to create.
Yep.
Early on.
Yeah, no lens, no cornea.
Whoa.
So it kind of looks like a piece of pepperoni. Like it's just
It looks like a button.
Yeah.
Kind of like a button. I don't know.
It's like almost kind of concave, fleshy. It's very strange.
Oh, cephalopods. There's a cephalopod eye. Here we go. Yeah. They're very eye-like.
Huh.
Like, I don't know. It's a little uncanny valley, these squid eyes.
Yeah.
There's something.
Yeah, it's one of those things where a fake cephalopod kind of looks more real than a real one.
Yeah. Yes, squid eyeballs.
It's,
Yes.
is it cake, but is it an octopus?
Oh man, that would be a great. Is it cake?
Yeah.
Is it a squid or is it cake
Is this a lump of rocks or an octopus?
or an octopus?
Is this seaweed or an octopus? Is this a coconut?
Googly.
Oh, octopus.
Octopus. It's an octopus in a coconut. Yes. Tool use, right? That was years ago when that coconut example came out. That video of an octopus scooching along the bottom of a little bay, carrying the coconut shell over. Like, what is happening? You're going coconuts. Cephalopods.
Cephalopods and frogs. They make my life worthwhile.
Oh, and so many other things. So many wonderful animals. And just, yeah.
The other day I was like, what if I did a whole calendar of just frogs? How would people feel about that?
Ooh. Some
There's a
people would
lot.
probably love it. I'm
There's
sure.
a lot of frogs out there.
Oh, Aaron Laura, I don't think I can. Squid Eye Molecular Secrets Smart Lens. Ooh, what is that link? Let's see. Cosmos. Oh yeah.
Oh, where was the thing? Somebody was talking about Italians and why people can't fit into a Fiat. Yeah,
I saw that.
Which species became Italians? Have you ever driven a Fiat? You can never get the clutch, pedal, and steering wheel positions to fit an American. Seems you need longer arms to make it work. So first of all, I just want to point out many Italian heritage people are Americans.
Yeah.
Just to clarify. But I had to laugh because that is, at least I always heard it in the family as being a Sicilian thing. Maybe it's larger an Italian thing. That we're short-legged people. So you got the upper body and the arm length and everything as somebody who is 6'4", but you got the leg structure of somebody who's 5'4", and then you end up somewhere in the middle with the actual height.
Yeah.
But Italian. At least this was the trope in the family. Although it didn't apply to somehow my uncles all ended up with the American legs, apparently. I got the straight up Italian body. I sit about 6'4". I sit tall, and I stand up, and I didn't really change that much. The legs
That's it.
are like, 5'1", 5'2", I think, if from the hip down. But the other, anyway.
I'm the opposite. I think, I'm the opposite. I have tall hips and longish legs for my height, and my waist is like this long. I have the shortest waist in the history of humanity. It's like everyone went trunk, squish, squish. So I should be taller. I am convinced. I got the opposite of the Italian. I don't know if that's the Scandinavian thing. I don't think so. But I don't know.
Yeah, maybe.
Oh, we're going. Are you both ready to do what?
I remember. Sorry, we can move on. But I just remember there was a teacher. This is like in junior high school or something. It was telling me to pull up my pants, that they were sagging because they didn't want people sagging their pants at school. And I was like, no, dude, this is my body. This is where this is just my body you're telling me to correct at this point.
I can't do it. Sorry.
They're up.
Sorry there.
They're not. There's no place that they're going further up either. I will tell you that. There's
Unless you
no-
Urkeled it.
Yeah.
They're not. Oh, skeletal dysplasias. Yes.
I'm all torso.
There's a bit of
Which
those.
I was telling the story. Like I had, this physical therapist once told me I had an unusually long abdomen, and I bragged about it to a good friend of mine who was quick to point out that's the nicest way that somebody can tell you have short legs. Otherwise they just say you're tall or that you're long. But if they separated your body into two
Long.
halves, it's
I
hard
can't
to-
imagine going to a doctor being told, yes, you're in the 90th percentile for length.
of abdomen.
Right.
Of just
What
the
does that
abdomen.
mean?
You're a long human.
We've measured you from tip to tail. You're a
Gross.
long
For some
human.
reason,
Well,
that's
well,
gross.
Great length.
it was a physical therapist, so I suppose at that level they're still like, oh, your body proportions are like this, and so here's how your leverage is when you're standing or leaning or
Yes, just normally when
Depends.
we talk about human length, it's called height. That's the only
Right.
thing I was talking
yeah.
about.
That's what I think my friend was pointing out is the fact that she had separated one half of my body as being unusually long, but left the rest of it completely, specifically even out of the
Leave it.
picture.
Leave it out.
Meant that it was really another way. There was another way to interpret that information.
Excuse me. Boobity boobity boop.
Share it together.
What were you going to say, Kiki, before?
Oh, no, I was just going to say, are you both ready to host the show on your own next week?
Yes.
Yeah, we got to
Do it.
remember that, Blair, because then we have to bring
Oh,
extra stories.
I got it.
You got it? Okay. Well then, if you've got all the extra stories, I'll just bring the huge.
You're welcome to bring one or two more. I will bring
Okay.
one or two more. It'll be great.
Okay. Yeah. Well, it
Yeah,
should be.
I'm
it should
gonna-
be,
I'm going to set up the 29th run sheet for
Mhm.
you so that
Mhm.
I'm not in it. And then it'll be easy for you because I won't be in it. And you'll go, what? Where? Oh yeah, Kiki's not here this week.
Mhm.
Okay.
Mhm.
And then everyone out there, the not Tuesday. We are a Wednesday show. The Wednesday. We do the show on Wednesdays. We have for years. Okay. Well, the Wednesday show after the 4th of July, we are going to be taking the week off. There will be no show on
The
You know,
6th.
on the 6th.
terrible. I feel like I rushed the disclaimer today.
What?
I feel like I rushed the disclaimer at the beginning. of
Really?
the show. Yeah. Because it was supposed to. I actually thought I was going to get interrupted. And I was like, should I keep, like, I was writing, I was like, should I just keep on writing this? Like, adding more things to it so that it would have the sense that it wasn't going to end. And it was a little bit provocative. I was going to see, like, what is the question? Get to the end of why does it keep going? But I think because I
was actually-
talking fast because I just had coffee.
No, you weren't talking fast.
slowed
Like
it
you started
down.
off at your normal pace and then you actually did take some pauses and I was like, what's happening here? And then you kept kind of going and in my head I was like, what question? And I was looking at Blair's face too. Like both of us were just kind of like, where is he going with this?
I had this idea that I would
So
write it.
definitely we were there in that space for sure.
Okay, cool. Because I had this idea too. I was a little disappointed. I was like, I should have just kept writing on it. Because if I was just like, what would happen if it just
Just
and that
keep going.
question that we all have had deep in our hearts and our minds in the moment we were conceived.
I thought you were just going to say it was the word why by itself. And then you started with huh. And I was like, oh, that's not what I expected.
Well, I said it was a string of words in a phrase a post-observational inquiry statement. But yeah, it was fun.
Huh.
Huh?
Huh.
Yeah.
Why is it like
What?
that?
Right?
What the heck?
Huh?
Huh?
What the heck?
Huh?
Woody hoo!
Hooty hoo. Hooty hoo, man.
Hooty hoo! Yeah, so Wednesday the 6th, no show.
And then all three of us will be back the week after that.
Most hopefully, yeah.
What?
Well, I mean, you never know. I didn't see you balancing on one foot.
Me? I was the whole show.
Oh, I didn't realize you. Oh, you've got this.
I'm sitting now. I stand the whole show and then I sit in the after show.
Oh, see, I didn't even.
I got
really...
a stander too. I've been standing.
Yeah.
Really?
Yeah.
Oh,
one
it's
foot,
me. I'm
and
the
then
one. I've
the
been
other
sitting the
foot.
whole time.
Sitting
Yep.
kills,
So
as we know.
Yeah.
I got my sit stand at home. I got my sit stand at work. I'm good.
I mean, standing's overrated though. I mean,
It all feels like
really,
though.
it's tiring.
By knowing
You
that,
really have to stand here any longer.
I find it the
You know-
opposite. I get sleepy when I'm sitting.
I have more energy standing.
Mhm.
Makes it better.
Yeah, I almost always start yawning immediately after I sit. Here we go. See, I said yawn. Sorry.
Itch, scratch,
Sorry.
scratch.
Oh, don't remind me. Gosh,
Scratch.
it's gonna be. I'm gonna need the week off just to forget that face mites are a thing.
Okay, but not next Wednesday.
Not next
No,
Wednesday. Yes,
The
you
one
both will be
after.
back next Wednesday.
I don't know
Next
if I'm gonna recover in
Wednesday.
time now for next week's show. I don't know, cause like it's a, and it's not. I know it's not. I know it's a face mite, not. It's but now like my whole head.
Right, it makes everything
It's-
itch. Right.
It's
Mhm.
like I just want to like
Yeah.
So I'm gonna
we
go
have
get some
other
sandpaper
scalp
and take
mites?
the dermal down a few layers just to see if I can shed some. But I guess the rest of the time I haven't
there?
noticed that I had face mites living on me. I guess I shouldn't really complain. They got rid of the things that make them active, it sounds like, at the wrong time, so they won't be noticeable.
Oh, Blair disappeared herself.
Hmm.
She's looking for face mites in the mirror. That's why she turned off her camera.
No, I want them there.
Yeah, you weren't there. You were looking for face mites. I know it.
Here's one Justin will want to compare. The length of the head and the length of the foot are equal. Oh, you gotta try that one in the after show.
Is that
The what?
like that joke of like, oh, your hand, if your hand is bigger than your face?
But you have to be in person for that one. There's the one where you can't put your elbow in your ear. And then the one that I fell for, my daughter got me. She's like, it's absolutely impossible for a human being to lick their own armpit. And I after having tried to put my elbow in my ear and failing, I was like, oh, I didn't that one. It seems like you could do it, and apparently you can do it. It's very easy, actually.
Yes. Yes, you can.
Yeah.
Yes, you can.
Yeah.
My face mites do not approve. That's all I have to say.
Your face mites don't want to talk to your armpit mites, okay?
No.
No, don't invent, don't make up body mites that don't exist.
Arn lore, generally body
adding
proportions are
more.
there are general rules for kind of body proportions. And like the foot and the head and the height, like there are like proportions that generally stand, but they're not always that way.
I don't think I'm
But I
really
mean,
gonna have
you got to get the foot up there. Like you're like, hello, this is the foot phone. I don't think I can do that right now.
No.
No.
Perhaps in the height of my yoga days, I could have done that, but
Pick
not
up the
anymore.
foot phone, please. No. No. Hello, Cappy. Meow, meow. Hi, meow, meow. We have a feline visitor.
All right, then.
Okay.
Hey, say goodnight, Blair.
Good night, Blair. Say good night, Justin.
I can't.
Say good morning, Justin.
Good morning, Justin.
Good night and bon
Good night.
voyage, Kiki.
Yeah, have a nice trip.
Thank
We'll see you in
you.
2 weeks.
Good night, everyone. Thank you so much for joining us. We always love it when you're here for the science with us. We love to be here with you. We will see you. Well, they will see you next week. Excuse me. Have a wonderful week. Stay safe. Stay healthy. Stay curious. Ha ha. Huh? Why? Huh?